Okay, so I want to talk about the 2A, 2B steps that I call them of cellular respiration. So we're looking at, first, we'll look at the oxidation of pyruvate. Okay, remember oxidation means lost, right? Loss of electrons or electron density. So basically we're going to be saying that in the end, pyruvate is going to lose electrons or electron density. It means it's going to be, electrons are going to be extracted. And basically remember, cellular respiration, photosynthesis, it's like all redox reactions because it's just transferring electrons. You know, so it extracts electrons, embeds electrons, extracts electrons, embed electrons. Electron transport chains involved and all that kind of stuff. So these are all, oxidation and reduction happens in many steps, not just this one, but I just call this oxidation of pyruvate. Some people call it processing of pyruvate, either way. So let's start with pyruvate. 3-carbon compound. I am keeping it simple with these. I'm not necessarily drawing the carbons how they're attached together. Usually I'm just drawing them in a line. That's what these things mean. They mean carbons, and of course they have oxygens and hydrogens and stuff attached to them in the molecule, but we'll just go with this. This is the extent to which you need to understand these processes, so I'm trying to just let you know the level of detail. Remember that every step we're doing here involves enzymes, so none of them just happen. There has to be enzymes that catalyze those steps by binding the substrates in the active site and then catalyzing the reaction and releasing them. Okay, so pyruvate is in the cytosol where it was formed after glycolysis and then ends up in the mitochondrion. Mitochondrion single, mitochondria plural, and it's actually has to go through transport proteins because can't just diffuse across the membrane. Because remember, mitochondria and chloroplasts are examples of membrane-bound organelles, and so they have phospholipid bilayers. Just remember there's transport proteins. And really, we're looking at this processes after it gets in there happening in the matrix, the mitochondrial matrix. Okay, so once pyruvate gets inside, the whole thing here of why is this happening, there's a couple reasons oxidation of pyruvate is useful. One of them is to make the molecule reactive. So in order to do that, remember one of the key points is you can make molecule reactive. Usually that means unstable. Okay. So you'll see. This is the general idea. So you'll think of it. Switch it out. So we're going to make a molecule reactive or unstable, and then you switch out that group that's making it unstable and add what you want to, whether it's electrons or another molecule to it, other types of groups, and those things. So that's a basic strategy. That's how phosphates are used to make molecules reactive. In this case, though, we're going to add the CoA to it. So we'll do that first to make it reactive and then switch it out. Yeah, switch out the CoA for what you want, which is to get into the citric acid cycle. So to do that, we're going to break a bond and bump it off. So you're going to break this bond, bump off that carbon, and it's going to leave as CO2. Add the coenzyme A, so here's your 2, and I'm going to make it a wiggly bond, and actually make it red even. So why did I draw this like this? This molecule that you've made here now is called your acetyl-CoA, and then that is ready to go to the citric acid cycle. Let me also show you here why acetyl-CoA is as reactive as it is, and that's because of this coenzyme A that's added. So here's your CoA. It's this entire huge group here. This whole thing is your coenzyme A. And then your acetyl group, your 2 carbons, it's just this tiny group here. Just this little group there. There's your 2 carbons. Those 2 carbons are what you want to add onto the starter molecule of the citric acid cycle. So in order to make that reaction happen, the bond needs to be unstable and reactive, and that's what happens here with the addition of that CoA group. So this is a reactive spot, and that means that basically the reaction can bump off the CoA and add those other carbons, and then you're able to get the acetyl, you can get the carbons from the acetyl-CoA into the citric acid cycle. But you can see this pulls a lot of electron density off of those carbons, making it very reactive. Another thing that happens is a second big idea. Break a bond, extract electrons. So break a bond, and then it's not really, you can get the electrons out, but during the process of breaking that bond, electrons can be transferred. Because remember, the bond is just the electrons being shared between molecules. These are all covalent bonds. So you'll notice here we broke a bond, right? That's what we were doing, breaking a bond. So what does it mean? Break a bond, extract electrons. Who's going to take those electrons? Your electron carrier. And so in the case of all the cellular respiration stuff, it's your NAD plus. The plus means that it's empty. It's like the cab with its light on. So it takes those electrons because you broke the bond, transfers 2 at a time, and then that makes NADH, which has the electrons in it, and that goes to the electron transport chain. From this process, you have acetyl-CoA, and then you have your NADH and CO2. CO2 just goes diffusing out the mitochondria, simple diffusion straight through the membranes, the phospholipid bilayers, and then can diffuse out of the cell as well. So it's a waste product here. Just make an inner and outer membrane to show you that this is the matrix where this is involved. So it's gone through both membranes. And I think it's actually 2 separate transport proteins, not one that crosses both. But for our purposes, this is okay. All right, so that's oxidation of pyruvate.